Good morning, everyone. We'll go over a short introduction and some of the logistics before getting straight into how to find and analyze influenza data in the BV-BRC. My name is Anna Maria Niewidomska, and there are several of my colleagues who are with me who are going to be helping me answer questions in the chat. So if you do have any questions, please go ahead and ask them. And we can also have some time for questions towards the end. We'll start off with talking about the different data types that are available in the BV-BRC, whether it's genomes, proteins, or other types of data. Then we'll move on to the services and tools that you can use to analyze this bioinformatic data on influenza. And then we'll take some time for questions and just have an open forum, and we can walk through how to do things if people have specific questions. And of course, this is being recorded and will be available on our YouTube channel after this is over. If you are new to the BVBRC and are not sure who we are one of the Bioinformatics Resource Centers for Infectious Disease that's funded by the NIH's NIAID division. We're established in 2004, and our goal is to basically provide free and public access to both the data and computational tools so that you can analyze the data for different kinds of genomic data relating to category A through C infectious diseases. And there are two BRCs. One of them focuses on bacterial and viral bioinformatics, and that combines what used to be the influenza research database, the virus pathogen resource, and Patrick, which is a database that focused on bacteria. We're now all combined into the BVBRC. VUPathDB, on the other hand, focuses on eukaryotic pathogens and hosts and vectors. So this is our new website. You can find it by going to bv-brc.org, and you can find all your viral information, and especially influenza, just by clicking on viruses. This will take you to our landing page, where you can either select the Orthomyxoviridae virus family that contains influenza viruses, or you can go to our featured virus list over here, which has influenza A virus, which is probably the most commonly studied influenza virus. If you're interested in other viral families, we also have them divided by Baltimore classification into single-stranded positive sense RNA, single-stranded negative sense RNA, and so on and so forth. And then down here we have, all the way to the right, our featured virus list, where we allow users to work on viruses that have been the subject of recent outbreaks. You'll see up here the different tabs that are available for your viral family that you're browsing, including taxonomy, genomes, features and proteins, protein structures, domains and motifs, epitopes, and experiments. And so these tabs might change as you navigate from one page to another or one virus family to another. And we can go through them and what they mean, but it's a good way to kind of explore the website. So today I'll be going through a few different things, especially nomenclature and structure of the website, because this is coming, especially if you're coming from IRD, the interface is going to be a little bit different from what you're used to. So understanding how the data is presented, what the different views are, and what the different wording is, and nomenclature for different things is important. So one of the main things that the BVBRC does differently than IRD is that it allows users to view data through multiple different methods called views. So you'll have the Taxon View, for example, where all genomes related to a specific taxon level can be viewed. So let's say everything underneath the Orthomyxoviridae family. You'll have the Genome View, where you can look at and explore data on a single genome, as well as everything else that's associated with it, whether it's metadata or links to other databases. You have the Genome Group View, which is a table that lists all of the metadata for a group of genomes. And then you have the Feature View. Now, for viruses, most of the time, the features are proteins, but you do have, or genes, but you do also have a few other things that might be things like 5' UTRs or RNA structures that might be important for that specific virus. And then the feature group view is the same thing as the genome group view, except it's just a table with a list of these. And we'll go through these one by one so you can have a chance to get more familiar with them. So today we're talking about influenza viruses, and these belong to the Orthomyxoviridae family. So one of the things I wanted to point out that's different from IRD and Viper is that IRD focused just on influenza viruses. So you had influenza A, influenza B, and C and D only, whereas the BV-BRC, if you're interested in other Orthomyxoviridae family viruses, you can find everything relating to this particular taxon, which includes the Corangi virus and Thuct viruses and so on and so forth. So, as I mentioned, there's the Taxon View. And so this is what it looks like. And again, I'll show you how to get there through the website as well. But once you click on the Taxonomy tab, this will take you to a page where you can browse through the taxonomy for that particular virus family. And if you're new to a viral family, I find this really useful because then you can understand the ranks, the taxonomy ranks of this family quite easily and understand what's a species, what's a genus, what's a family, and so on and so forth. And once you've selected the particular taxon that you're interested in, let's say just influenza A virus, you can click on the genomes, which will take you to a page that just has the genomes for that particular virus, or features, which does the same thing, or you can click on Taxon View, which will take you to the same Taxon View, but just for that particular virus. So if I was going to do that on the live website, and I'm guessing you guys can see my screen now, you would navigate to bvbrc.org, click on viruses, and then this takes you to our virus landing page where you can just go ahead and click on Orthomyxoviridae. And this takes you to the Orthomyxoviridae landing page. And this page just summarizes everything that we have on the Orthomyxoviridae family. It tells you how many genomes we have, how many proteins, how many mature peptides, how many protein structures, and gives you a little summary of what's there. And then switching to the taxonomy tab, as I mentioned, will take you to this list of influenza viruses. Just to be different, I'm going to select influenza B over here. And then if I switch to a different tab without pressing on Taxon View, that's going to take me to the tab for all of the Orthomyxoviridae. But if I click on this Taxon View, it's only going to select for influenza B virus. And you do have this kind of breadcrumb trail up here, which will give you a fairly good idea of what it is exactly that you're looking at. So now when I go to the Taxon tab, it's only listing viral strains that belong to influenza B. So then we have our strains tab, which allows users to visualize and select genomes that have all of the segments sequenced. And this is really useful when you're looking at segmented viruses, which obviously are going to have multiple genomic segments. And navigating through GenBank or NCBI virus can be a little bit frustrating because you don't really know which segments belong with what. But if we flip to the strains tab over here, you'll see that each column over here is labeled by the segment name, 1, 2, 3, up to 8. And it shows you how many segments through the segment count column are available for each one. Now, for some of them, we have less than 8, which is the expected number of influenza segments. Some of them are complete, and some of them are partial. And some of them have more, which means that they've been sequenced multiple times. So there's often multiple passages, and they get sequenced as they're passaged along. And you can filter by clicking on that filter button using the metadata that's available. And you can choose whether you want a particular subtype, Victoria or Yamagata. You can choose whether you only want, let's say, complete genomes. You can choose whether or not you want a specific host or a specific country or a specific collection year. And these are not the only fields that are available. So you can click on this little gearbox and customize the different selection options that you're interested in. And once you've done that, let's say you're interested in just looking at complete genomes that were sequenced, let's say in 2020. So this will give you 535 results. If you flip to another tab, such as the features, which will show you the proteins, or the genomes, which will show you the nucleic acid segments that are associated with these records, then it will take you back to the entire list for influenza B viruses. If you would like to apply these options over here, then you can do that by selecting the metadata that you want through the filters. So again, we'll click on, let's say, everything that was collected in 2021. You click on the apply button, and this will change your breadcrumb list to show you that collection year is selected. And then as you flip through the tabs, the features again will have this metadata criteria selected. So going back to our presentation, this is our other view that I was talking about, the genome group view, which allows you to see all of the records that are there for each segment. So over here is just a screenshot of influenza A, for example, and you have the different metadata that's associated with each record. And we can take a look at what that looks like over here on the live site. And I also want to mention that all of these columns are customizable. It's a little bit hard to see, but there's a little black button with a plus sign over here. And if you click on it, this is going to give you access to a treasure trove of information that's associated with all of these records, whether it's on taxonomy, whether it's on the typing information, let's say the H1 type, the H type, for example, or the N type, so that you can choose whether you want H1 or N1. Obviously, this only applies to influenza A viruses, not B. Genome quality, whether you want information on cross-reference databases. Let's say you want any information on publications that are associated with these records. That will give you a column over here with that information in it. And then isolate information and host information, which is extremely important. And these include things like the collection date, the geographic group. So one of the things, for example, that people are always interested in is not necessarily looking at data from the United States, but from all of North America, including Canada and the US. And the same goes for other geographic groups, which include Europe, Africa, Asia, and I think, what else do we have? Yeah, the major continents, the major WHO geographic groups. And then the host information, which is also extremely granular, sometimes can include host age, gender. We also have included and curated our information by host groups, which includes things such as mammals, avian species, sea mammals, insects, if you're interested in vector-borne diseases, and so on and so forth. So I really encourage you to kind of just click through these and explore what's available, because in some ways there's a lot more here that you can go through than you could in IRD. So, so the genome view. So once you've found your particular genomic segment that you're interested in, you can click on it and go to this tab over here in this green action bar and click on Genome. And this will open a new tab that takes you to the Genome View and shows you all of the information associated with that particular record. And then you have the different tabs that apply only to this influenza B virus from Alaska, including a genome browser, the features and proteins, whether there's any protein structures or domains and motifs associated with it, and so on. Next, we've got the feature group view. And so if I go back to my window here and switch to the features, this is going to show me all of the information for those influenza B viruses collected in 2021. But it's going to show me the genes or the open reading frames that are available for them. And so we can see all of the different proteins over here, the HA, neuraminidase, PA, PB, PB1, PB2, etc. And you can filter these. And we have two different flavors of annotation. We have our own annotation, and we also have annotation that's imported from NCBI. And one other thing I wanted to point out is that you can search for almost anything by clicking on the advanced search. If you have too many records, you can filter it out by length, by what particular gene or product that you want, by taxon ID. It's a really good way to kind of pin down exactly what you want from your data. And then similarly, you also can look at what's available for an individual record by selecting your record and clicking on this feature view, which will take you to all of the information associated with the proteins that are in that genomic segment, in this case, M1 and BM2. So how are we doing on time? All right, so next I thought I would just go through the different things that are available in these tables, and I think I've covered most of them. At the top, we do have these breadcrumbs, which will take you to, which will show you exactly where you are. And then we have the download button, which will give you the option of downloading all of the information in this table. So you can just select everything that you want by clicking on this top button here, or you can just select the particular records that you're interested in. And one thing I didn't know about is that you can just use the shift key to select more than one record at a time. You don't have to keep pressing every single button. And then once you click download, it gives you the option of downloading this table either as a text file or a CSV file or an Excel file. And you can also look at the individual FASTA sequences, and this gives you the option of viewing them either as DNA or as proteins, which is really useful. We also have the keyword box, which is a really powerful way of searching for what you want. Let's say you're only interested in any entries that say Alaska in them. And you can just use the keyword search to further narrow down your results. And it turns out that only 132 of these records have that keyword in them. And I think that's not something that was available in IRD previously. We've talked about the filters that you can access by clicking on the filter buttons, the column headers, which will obviously tell you the different types of metadata that are available for those entries. By clicking on them, you can kind of you can sort through them. So let's say you want the longest segments at the top. You can just click on these and it'll sort through them for you. On the bottom left over here, it'll show you the total number of results that you have per page, how many pages there are on the page count, the column selector so that you can customize your table, and then the row selector so that you can choose the individual entries that you want. And then over here in this green action bar, these buttons tend to change depending on what page you're looking at. But they're really useful, so I would encourage you to kind of mouse over them so that you can kind of understand what they do, and just hovering your mouse over it will give you a little explanation of what each one does. And then finally, we have a little information panel so that if you click on a specific entry, you can view the information that's associated with it in this little panel side by side. And so this is, I briefly mentioned the action bar. We have, again, different buttons that have different functions. So we have general purpose ones, the hide and the show button, which will allow you to close and open that panel so that you can make your table even wider and fit even more metadata into it. The download button that's available there for on most pages. You can copy rows. And then this group button, which is incredibly important because this is how you're going to make your working sets that you want to analyze for any of the tools on the web page. So you can, let's say we're going to select these first few entries over here, and I'm going to go ahead and click on group. Let's say I only want these PB2 proteins at the top. And I'm going to click on group. And what this is going to do is open up this window that tells me, do you want to save your data to a group? And so there's two different types of groups on the BV-BRC. One of them is called genome group, and that's how you're going to save all of your nucleic acid sequences. And the other is called a feature group, and that's how you're going to save your protein sequences. And so the BV-BRC comes with two different folders that exist as a default. And those are the feature group folder and the genome group folder. So in this case, it's proteins, so I'm going to keep feature. And I'm going to say that I want a new group. And it's going to save it by default to my feature groups folder. And I can just give it a name here, Influenza B, BB2, and click Add. And so now I can use this in any of these tools and services that we'll get into later on. So let's see, what other buttons do we have in the action bar? We have the feature button, which I've gone over, or the features, which will show you the entire table for a specific group of entries that you select. So let's say, for example, I'm looking at some influenza genomes, and I select these first 6 entries. And these are genomic segments, but I want to know what features are there. I can go ahead and click on this Genomes button, which will take me to another page that just includes these. And then if I switch to the Features tab, all right, we can come back to this in a little while. Let's see, what other buttons do we have? We have this FASTA button, which will allow you to view the FASTA DNA or protein sequence associated with an entry. ID mapping, which provides the option to map different identifiers, let's say the GenBank identifier with a PDB identifier. This button that allows you to generate multiple sequence alignments. Let's say, for example, we go back to our list over here of features, and you want to align a couple of them. You can just select them and click on the multiple sequence alignment button. So sometimes your best bet is to just refresh the page if there's a lot of traffic, or you can just wait patiently. In this case, I'm going to move forward, and you'll just have to trust me that selecting multiple entries and clicking this multiple sequence align button generates multiple sequence alignment. You also have different buttons that are available for your workspace, very similar to what you could do in IRD. You can share different folders, you can delete them, rename them, move them. And then something that's new over here is that you can change the file type. So let's say you can change TSV to a text file, for example, which will change how the workspace interprets and views the file and the tools that you can use it for. So if you're coming from IRD, you might have a little trouble figuring out how exactly to find what you want. There's a few different ways. One of them is just through browsing, which is what I've just showed you. The other is the global search, and the third is the advanced search. So I'm going to show you quickly how you would go about doing a global search. And you can do that either through this box that's available here, or if you're already in the viruses page, you can find it on the top right corner. And if you want, let's say influenza C virus, you can just type in influenza C. If you want a specific data type, let's say you just want strains, which includes all of the genomic segments available for influenza C, or just the proteins, or just the genomes, you can click on that. But I like to leave it as all data types because it gives me a really good idea of everything that's available in the database that has the word influenza C in it. So this at the top, you have a summary box of what's available in terms of the taxa, the genomes, the different features, protein structures, domains, and motifs, immune epitopes, and you can browse through that and see the top matches by just scrolling down. So the third way is if you already have something very specific in mind, you can use the advanced search option. So how do you get to that? Going back to the web page, you can go to the second tab over here that says searches, click on that, and let's say you're looking for a specific protein or a set of proteins. You can put in a keyword, you can select your particular pathogen group from the drop-down menu, and this includes all of the featured virus families and featured viruses. Or if you have a specific taxon in mind, let's use influenza C, for example, again, as an example. And this will give you a drop-down menu of predicted names. And over here, this is the one that I want, the species influenza C virus. If you know the particular genome ID, you can put that in. And this is just our internal ID for the BV-BRC, but it probably won't apply for most viruses, although it's useful for bacteria. You can select a particular host group. If you look at our drop-down menus, these are our general broad groups that we have available. Let's say I want everything that was found in humans. You can give a specific host name, and there's a lot available here for you to browse through. The geographic groups that I mentioned before, let's choose North America, and a specific collection year window. Let's say I want everything from the past 10 years or so. And you can specify genome length, a specific gene, and there's a lot of additional criteria that you can browse through by clicking on this drop-down menu. And you go ahead and click search. And that should give you a list of the proteins that are available for that specific search query that you've entered. Now, if it says 0 of 0 results, don't be scared. Pay attention up here and make sure that it says it's loading, and those should appear for you. And there we are. We've got 69 influenza C proteins that were isolated from humans in North America in the past 10 years. So what are the different kinds of data that you can find in the BV-BRC? So we talked about those a little bit already. We talked about strains, we talked about genomes, we talked about features and proteins, but we didn't talk about protein structures. So if I go back to my homepage, click on viruses, navigate back to my Orthomyxoviridae, you'll notice that there are a lot of different tabs available here, and we didn't necessarily go through all of them. And this can be a little bit slow sometimes, but we'll get there, I promise. If you navigate to the Protein Structures tab, this will give you a list of all of the structural entries that are available in the database for influenza. And this includes structures that were generated through electron microscopy, NMR, or X-ray crystallography. And again, you can use the filters to search for the different things that you're looking for by the metadata that's available there. So let's pick a smaller one here. Click on the genomes and the protein structures, and that should open up. Once you have your table, you can select a specific structure and go to this action bar. And again, you'll notice that these buttons have changed. If you click on the structure, this will open up the protein structure viewer, which is also something new on the BV-BRC. We've changed our structure viewer to Moldstar, which is really beautiful structure viewer, allows you to interact with the protein structures in ways that you couldn't on IRD, and it's worth exploring. Next up, we have the tab next to it is the domains and motifs. And these are the different predicted domains. Let's say if you're looking for a particular structural domain or something like that, you can find it by clicking on domains and motifs. When that loads, you can click on it and view the particular domain. You can see the source where it's from, for which particular gene or gene product, and all of the information associated with the particular entry in the side panel. Of course, we also have immune epitopes, which were also available in IRD. And you can browse through those by clicking on the epitopes tab. That will show you B cell, T cell, and MHC class epitopes. You can browse through those, again, using the different host metadata, epitope type, whether you want a linear or a discontinuous epitope, and by specific protein, of course. And then we have surveillance. So where do you find information on influenza surveillance? So it's not listed in these tabs, but you can again find it by clicking on this search button and going to surveillance. This will take you to a search page where you can enter everything that you're interested in, which particular influenza virus, if there's any specific metadata, and that takes you to, that will generate a table. Over here, as an example, I have everything that tested positive for a specific species of duck in a host group of avians. Once you have that, you can select all of your entries and click on this map button in the green action bar. And what that does is open up a new page that will show you exactly where those samples were taken, which ones tested positive. And there's a lot of things to explore here. You can overlay, for example, the specific flyways or that show you the migration patterns of birds in those geographic areas. So a lot to look through. Same goes for serology data. You can find that again by going to the search tab, clicking on serology. You put in your input query, click on search. That will give you your results. Again, you can click on a specific entry, click on the serology button to look at all of the information associated with it. We also have host factor data, which was previously available on IRD. It's a little bit, it's not necessarily intuitive how to get to it on the BV-BRC, but we are working on that. And you can get to it by clicking on eukaryotic hosts. And this will take you to a table where if you click on the experiments tab, you'll be able to see the different omics experiments and the different experiments that were done, let's say, using human or mouse hosts with something like influenza. Over here, for example, we've got an H1N1 mock infection, and that should give me the results over here. But we've got chip data, we've got lipid quantification, protein quantification, and transcript quantification data. And I think we should probably have a tutorial on how to go through these and how to browse and visualize and analyze the different data, because there's a lot of valuable information in here if you know how to access it, but it can take a little while to know how to do that. So I'm going to pause and see if there's any questions before moving on to the tools and services that can be used for analysis of influenza virus. Tools and services that can be used for influenza. So we can divide these into a couple of different categories. We've got some new tools that are available for sequencing and annotation that maybe weren't there in IRD. We've got tools that are available for comparative genomics, and these are things that you're probably familiar with if you're coming from IRD, multiple sequence alignment and SNP analysis, phylogenetic trees, the Metacats tool. We've got the basic things like BLAST, primer design and subspecies classification for the different influenza viruses. And then something that might be new for a lot of people is the metagenomic tools, which include taxonomic classification and metagenomic binning. And so I'm going to go through all of these a little bit quickly, just to give you an idea of what the input page and output page looks like for each of these tools, but we'll leave some time for questions at the end afterwards. And then just to compare the workspace versus what was known as the Workbench in IRD, I should mention that you do have to be logged in to use these tools, and so that you can upload your data to the workspace. But if you are a previous IRD or Viper user, your credentials should still work for this site. So you can just log in with the same ID and password. And using this workspace, you can do the same things that you could do in IRD, including upload and download your own files, share them with other users, copy them, so on and so forth. But I do want to, again, drive home this idea of the genome groups and the feature groups, because these are going to be the foundation of everything you do in the BV-BRC. The genome groups are where you're going to upload your nucleotide data sets, and the feature groups are where you're going to upload your gene or protein data sets. And you can get to your workspace by navigating to the Workspace tab up here, clicking on that, and going to your home folder, and that will show you all of the different folders that you have, including your default genome groups and feature groups folders. Let's start with FastQC and assembly. For anyone who's done next-gen sequencing, you know that FastQC is a really great way to kind of get some quality control information on your next-gen sequencing run. It'll give you an idea of how good your run was. And then for our assembly, right now we only offer de novo assembly, which can be useful for especially amplicon-based sequencing of viruses. But we are also working on reference-based sequence assembly pipelines, and we're hoping to have those available in the near future. But you can use our de novo sequencing assembly to assemble your viruses as well. And you get a nice HTML report at the end, which gives you a nice bandage plot to visualize what you have assembled, some mapping statistics, and some assembly statistics as well. Once you have your genome assembled, you can annotate it in the BV-BRC, or if you prefer to assemble it through a different pipeline, you can still come and use our annotation service. And basically, everything that you want can be found under our Tools and Services tab. So if we go to Annotation, that will take me to the annotation page that's shown in this slide. And it's quite simple to use. You just upload your file or browse for it from the workspace. You make sure you select the specific annotation recipe that's relevant. Obviously, for bacterial users, they're going to want to select bacteria or archaea or bacteriophages. But for people interested in influenza, you should select viruses. Select the specific taxon that's relevant to you. This example shows influenza A. Give it a name. Make sure you specify a specific output folder for it, and then just click on Annotate. The annotation results will come up, and you can find those by going to your jobs, which you can find by either going to this tab at the bottom right corner of the page, or by going to the Workspace tab and clicking on My Jobs. Let's say you have an annotation result. You can double click on it, and that will take you to the job results page, which has sometimes a long list of output files that you might not be familiar with. But there are tabs over here to help you visualize the results. Let's say you just click on this I button to take you to allow you to visualize your annotated genome. It'll give you an overview of what's there, allow you to look at it through a genome browser view, and then you can click on the features to look at the individual proteins that were annotated. And something that's also important is the output results include GFF3 and TBL files that are sometimes necessary if you want to upload them to NCBI. Oh, and I forgot to mention that we have specific annotation databases available for influenza A, B, and C, and we are working on influenza D, and that should be something new that was not available in IRD. Taxonomic classification. This is a fantastic tool that, let's say you do whole genome sequencing of a metagenomic sample, such as a nasal swab or bronchio-alveolar lavage, and you can see what's in your entire sequencing sample by uploading your file, or you can even throw in an SRA run number into there, which is what I've done in this example. And it'll give you an idea of how many reads are there for each specific taxon. And you can see in this example, we've got influenza A. And I know I'm going through these a little bit fast, but I just want to make sure we have time for a discussion and questions at the end. And I'd be happy to go over these again individually for anyone with questions. We have metagenomic binning. So if you do find a sample that you're interested in, but let's say you're not interested in the 90-something% of host reads or bacterial reads, and you just want the 0.1% of viral reads, you can use metagenomic binning, our metagenomic binning service, which will separate the different reads into different bins that you can then go on and assemble into contigs and further analyze by using this service. So in this example, this is a result of the virus binning summary. And I used this previous example of this SRA run over here, which contains influenza A virus. And so over here, I have the different reads that have been placed into each of these different bins. So I have my influenza A virus, similar to this Puerto Rico strain, and influenza A virus, similar to this New York strain over here, and this mystery virus in the third bin. But it's a great service, and I encourage you to explore it and see what you get. If you're not busy over the weekend, it's fun to just dig through SRA sometimes. Our multiple sequence alignment and SNP analysis. This is pretty self-explanatory, but it's a single service over here, whereas in IRD and Viper, they were separate services. But basically, you can go to protein tools, although this can make multiple sequence alignments and look for SNPs in genomic segments, of course, as well. You have the choice of whether you want to upload unaligned or aligned sequences. You can include either DNA or proteins. You can include feature groups, which are the saved protein or gene groups, viral genome groups, which are the nucleic acid ones, DNA, or upload your own DNA or protein FASTA file. And then this feature, which is new in the BV-BRC, which is really nice, you can choose whether or not you'd like to select a particular reference sequence. And you can either do that by selecting a specific feature ID or by copying and pasting your own input reference sequence over here. We have a new aligner as well. I think IRD only used muscle, but over here you can choose to use MAFT as well. And then you just specify your output folder and your job name, click on submit, and then you can find your jobs by clicking over here. Right. So, phylogenetic tree building. This is something that can be found by going to the tools and services and clicking on gene tree. There is another service called, let's see, where is it? Phylogenetic tree over here. But this is just for bacteria. As you can see, it has a B next to it. Services that generally apply just to bacteria have a B next to them. Services that are specific for viruses have a V next to them. But if you'd like to use our tree builder, you can click on Gene Tree. This will take you to a page where you can upload either DNA or protein files. You can use saved feature groups or genome groups from your workspace. You can add multiple file types. Let's say, what shall I choose? Let's say I'm going to pick some HA files that I have over here and click on plus to add it to the selected feature table or file table. And let's say you want to add another one. You can add multiple files that will be aligned and it'll generate a tree from them. There are a few different parameters that you can choose to adjust the ends or gaps in the sequence. You can choose different types of trees, including a couple of different maximum likelihood options and fast tree, which is a neighbor joining method. You can choose different models depending on whether you're selecting a DNA or protein phylogenetic tree. And then you just enter the data for your output folder, name, click on submit. The job result will give you a lovely tree that can be opened in our Archaeopteryx tree viewer. And then you can customize your tree with a ton of different lovely colors that are based on the host metadata that are associated with the entries in the database. So you can color it, let's say, by species, by host, by year, and it's a really nice way to visualize your trees. We have our Metacats tool, where you can compare different groups of genomes or genes. You can use this to look for statistically significant differences between different groups of genomes or proteins based on metadata. So let's say you want to compare the HA sequences that are circulating this year versus what was circulating in 2019, which is what's shown in this current example I have over here. It will give you the positions in your HA gene that are most statistically significant, as shown by the top p-values over here. And then this is comparing what's available in my 2019 HA sequences from September versus my 2022 HA sequences from September this year. So the most statistically significant position in this case is at 297. There's a lot more variability at this site in 2019 with some F, L, and M amino acids at that site, whereas in 2022, the majority of them are isoleucine and methionine. You can also use our BLAST service to look at our local databases. So you can get to that by clicking on Tools and Services, clicking on BLAST. You can input whatever your sequence is, either by selecting a specific FASTA file from your database, from your workspace. Let's say I'm going to click on this particular file over here. You can select a specific database of just viral reference and representative genomes. You can search within a specific taxon. So let's say you want to blast only against influenza A. You can do that over here. And then you just select your input and output. You get a list of the top matches, which clicking on, you can then view the alignment and save these as well. You can also design primers if you want to go on to perform PCR or real-time PCR experiments. And this is pretty self-explanatory, but I can also go through it if anyone wants more details. We have subspecies classification, which is in development still. We have a lot of things that have been that were available on Viper and IRD. So far, it contains subspecies classification for multiple different Flaviviridae. For influenza, we have an H5 classification and swine H1 classification. We are still working on migrating the H1N1 classification service over from IRD, and we are also planning to provide subspecies classification for RSV. So I thought I would end by comparing what's available in IRD versus what's available in BV-BRC, just so that everybody is aware of what's there. But I'll pause and just check if there's any questions before I do that. Okay, so basically pretty much all of the data that was available in IRD has been migrated over to the BV-BRC, whether it's strains or genomic segments. The one thing that I did point out was new was the taxonomy. So we include all taxonomic groups, including things like Corangavirus and the entire Orthomyxovirus family, rather than just influenza. You can still view our genomes through our genome browser. We do have animal and human surveillance data. We have human serology data. You can still find protein structures, host factor data, immune epitopes, and phenotypes. The one thing that's still not quite there is the phenotypes and the sequence feature variant data types. The domains and motifs are there, but we're in the process of updating the sequence feature list because it's over 10 years old, I think now, but that should be there soon. Services and tools. So while there are some things that are still not quite there yet, let's see. There we go. Most of the functionality that was available in IRD is there. So BLAST is there. Multiple sequence alignment in Viewer is there with the added ability to make multiple sequence alignments using MAFT. SNP analysis is the same. Phylogenetic Tree Builder is there. Sequence annotation is there, and we're also adding the ability to annotate influenza D as well. Metacats, PCR primer design, the structure viewer are all there, although the structure viewer is new. It should provide some additional functionality as well. The clade classification is there. The only thing that's missing so far is the H1N1, the human H1N1, and this should be there shortly. And so now this table summarizes what is still has not been migrated from IRD. And that includes the antiviral risk assessment, the identification of sequence features and segments, looking for specific point mutations, and the HA subtype numbering conversion. Those are have not yet been migrated from IRD, but we are updating them, and we do plan on having them there soon, and we will not shut down the IRD website before they are. There are new functionalities that I briefly went over, and I'm happy to expand on as well, including the FastQC utilities, the de novo assembly service, the taxonomic classification and metagenomic binning, and RNA-seq analysis service as well. In the meantime, if there is something that you really miss from IRD and you can't find it on the BVBRC, you can still use the IRD site by going to legacy.fludb.org. The timeline originally to shut down the site was March 2023, but we will keep the site up until we feel that our users no longer need it, and everything that you could do in IRD, you can do in the BV-BRC and more. The one caveat that I will mention is that the data is no longer being updated in IRD. So if you're looking for data types or influenza virus sequences or proteins, I encourage you to do that through the BV-BRC, not the IRD legacy site. In terms of data submissions, you can still do that through IRD, through the legacy site, and we are in the process of updating it. So for people who use our sequence submission service, you can still do that through IRD, but you should be able to do that through BVBRC shortly. And therefore, for anyone else who submitted other data types to IRD, including experimental data, surveillance or serological data, you can also email us and send us those things manually in the meantime, and we will upload them to the BVBRC. And then finally, for the big data users, comparing the IRD API versus the BVBRC, you have two different options for large data access through the BVBRC, and that's through either the API or the command line interface. You can pull incremental data based on the date, and you can use anything in the metadata to specify specific genomes or proteins that you're interested in. Anything that's available in the database in terms of metadata, you can use for your query. Things like seasons, hemispheres, geographic groups, host groups. And if you're having trouble, please do email us. We really encourage you, because this really helps us think of specific use cases that are going to be useful to you. And then we can put together kind of a cookbook and a list of example use cases that can be helpful to you and others like you. If you're interested in learning more about the BVBRC data API, you can find it by navigating to bvbrc.org/API/doc. And there is a very good user manual showing all of the different data types, all of the different metadata fields that can be used. And so these are just some examples. For example, where you can look for all of the genomic sequences for a specific taxon ID, or you can look for all of the specific proteins for that specific taxon ID, and let's say you only want the NP gene. And so that'll take you to, let's see, if I click on one of these links, that'll open up a page with all of this relevant information on it. The other option for using the BVBRC is through our command line interface. You can find more information on that by going to our documentation and clicking on these help resources and clicking on command line interface. This will give you a pretty good idea of how to download our CLI and how to use it, a few different use cases. You can also find more information on our GitHub site at github.com/BVBRC, BVBRC CLI. And then there's pretty much nothing you can't do on this site. You can use all of the tools. You can use all the different command line options to run huge batch programs and huge batch data sets through our different tools. So if you're someone who's more interested in running large data sets through these tools, please feel free to also get in touch with us and ask about our CLI if you have any questions that aren't answered through the documentation. And finally, I will end with just saying that there's a lot of information that you can find through our social media sites. For example, our YouTube channel has a lot of instructional videos and tutorials. We put all our webinars and presentations up there. If you want to stay up to date on our upcoming free webinars and workshops, you can follow us on our different social media sites, including Facebook and Twitter, although these days Twitter is questionable. And you can read more about us under our, by clicking on the About tab, looking at our information. And if you have any questions, just click on the Contact Us button over here. This will open up a box and say, hey, I'm having trouble finding, etc., or I can't figure out how to do this. And we usually get back to you within 24 to 48 hours. And that is it. I will pause for questions and see if we can demo anything specific.
Yeah, I thought that was great. There were some questions, but I think we covered most of them. You did, let's see, where you asked about metagenomic binning right before you did that. There was a question about the, well, Richard pointed out that the IRD GenBank submission service is going to be added in January 2023. And oh, here's one thing that there was a couple of questions on. Could you go back to like a genome table or a feature table? And there were some questions about the vertical green bar and how it's enabled. So if you could just select on one and show the icons versus two or more so that they can see what's there was some question about how to enable the icons in the vertical green bar. And it depends on the number you've selected, as you will show us all.
Sure. So if you select one entry, it's going to show you all of the information associated with that one entry. If you select a group of entries, that might change. And obviously, you can't see all of the metadata for all 10 items together. But if you select all of these, you can select to group them, or you can click on the Genome View list or choose whether you want something specific associated with these 10 genomes. So let's say if you want to just see the features associated with those, whoops, pressed the wrong thing. If you just want to see the features associated with these, you can press and hold it for more options, and then click on the features, which will show you just those proteins for those few entries that you selected. Okay. So it's worthwhile to just take some time and browse through the website, pick your favorite virus and explore, just so you can get comfortable with it. We know that it's going to take a little while. Whenever you change systems, it takes a little while to warm up to the new one. And we do expect some bumps along the way, but we hope the new functionality will more than make up for that.
I think that pretty much handles all the questions, unless anybody else has anything they would like to add. And you can always use the contact us, and we're ready to help you.
Yeah, absolutely. Nothing better than answering user questions all day.
We love that.
It's actually really, we really appreciate you guys when you send in questions, because then we can know how to help you, how we can improve the site.
Alexi Porollo asks, what tool is being used for metagenomics binning? In the tutorial, it lists the tool. It's by Perello et al. And it is our own pipeline. but we are contemplating adding additional ones. I go into tutorials, and I think it lists it right at the top for metagenomic binning.
I think we're also using CheckV for the viruses.
Yeah.
So if you go to this page,
true.
it'll give you an idea of what's used for the bacteria versus what's used for the viruses. So bacteria uses the BVBRC's own annotation pipeline, whereas the viruses CheckV is used. But you can read more about it, as Rebecca said, in the tutorials.
Also, if you scroll down to the bottom of the tutorial, if you use any of our services, we provide you with the references for you to use. Yeah, Alexi asks if there's a pub associated with it. That publication for the bacterial part is Pirello. That's the top one. And then there is another one for CheckV, but there it is at the very bottom. Where's that CheckM? No, that's for genome quality. It's hard for, there it is, NIFOC. So when we do these tutorials, what we plan on is we recognize that you run the services and then you may go off and you're writing your paper after maybe several months and you can't remember exactly what you did. We try to give you the information here so it makes it easier for you to craft your method section and your results section as well.
All right, well, if there's no more questions, thank you so much for your attention and attending, and this will be online within probably 24 hours. Thank you so much, everyone. Have a great day.